Now that we have an understanding of some of the important key terms that we're going to be using throughout this inheritance topic, let's have a look at Punnett squares and how we can use them to help us determine the proportions of different genotypes and phenotypes in offspring. So very simply, a Punnett square is a diagram that we use to show the possible combinations of alleles and genotypes of offspring. So I've got an example of a Punnett square here, and this Punnett square is used to show tall versus short, and we can assume that maybe this is talking about plants. So if I highlight my alleles here, I'm using a capital allele here for tall, and this indicates to us that it must be the dominant allele. Using my yellow highlighter on the other hand, my small t, my lowercase t, denotes the short allele, which in this case is a recessive allele. With our Punnett square, we can see here that we have two parents, and our first parent, which I'll highlight, has a heterozygous mix of alleles. Actually, let's switch that to green for capital T and little t. So our first parent is a heterozygote, and if they're a heterozygote for a dominant trait, they're going to have a phenotype of that dominant trait, so they're going to be tall. My second parent has the same genotype, they are a heterozygote, and as a consequence, they will also be tall. When we look at a cross between these two parents in my Punnett square, however, we end up with one individual, first of all, who is homozygous dominant, two individuals who are heterozygotes, like their parents, and finally, one individual who is homozygous recessive. Based on this information, we can then represent this as different ratios to show the different genotypes that are present in our Punnett square. The first instance, we can represent this information very simply as a ratio. So we have 1 to 2 to 1. One dominant heterozygous, one dominant homozygous, two heterozygous, one homozygous recessive. I could also represent this information in the same way, however, this time as a fraction. No one way is more right than the other, but it's important to carefully check the question to see what they are asking you to do specifically. Lastly, we have the percentage method. This is my personal favourite. I find it the easiest, but again, there's no right answer here. Please use what works for you. Now, our genotype is, well, our alleles. Our phenotype, on the other hand, is what is shown, what is displayed. So when we look at our genotypes and we look at these different ratios, we can see that we end up with 3 tall to 1 short plant. We end up with 3 out of 4 tall to 1 out of 4 short, or 75% tall to 25% short. Using the percentage technique, we know that every square in our Punnett square is going to be weighted at 25% each time. So some simple rules for us when we're doing a Punnett square involve, number 1, starting by drawing a square, almost like a game of tic-tac-toe, and then drawing that checker box inside. Step 1 done. Next, if we follow our alleles from our previous example, we write the alleles for one parent above the column. So capital T, little t. Step 3 is where we fill in our other parent, and our other parent was the same in this scenario, so capital T, little t. Step 4, we now write the combinations of alleles in this box here. So we have homozygous dominant, our heterozygotes in these two boxes, and lastly, our homozygous recessive individual. Number 5, and this is what we've just done on the last page, we can calculate the probability of each genotype and each phenotype. So, when we're working with Punnett squares, it's important that our working out follows a particular format. So to work it through before we go through an example together, first of all, we need to state our alleles. And when we're choosing alleles, please be careful to choose letters that are different in their uppercase and lowercase format. So some good examples of alleles which are different include things like A, include things like D, include things like F. Things that are not so good, okay, things that can be quite difficult would be O or S, or even though we've used it previously, T can still be a little bit confusing sometimes. Once we've stated our alleles and we've clearly stated what they represent, being either the dominant or recessive trait and whether or not, in our case, it was tall or short, we then let the marker know the parent phenotypes. So what does each parent look like? So in our last instance, it would have been something like tall and tall. We then have a look at the parent genotypes, our next step, and our parent genotypes in this instance were heterozygotes, both of them. We then simply copy these into our Punnett square, as we have done a few times now, and then after that we complete our Punnett square. We fill that in with all of our information. Then our second last step, we write those genotypes. And our genotypes again can be a ratio, they can be a fraction, they can be a percentage. And our last step, we represent the phenotype. And this is simply our statement about what the possible offspring look like. So what do we actually see? So to work through an example now, we have the following scenario. A pure breeding male white mouse is crossed with a heterozygous black mouse. In mice, black is dominant to white. Determine the fraction of white to black mice. Now in this instance, again, we start with our alleles. And in this case, we haven't been told a specific allele, so we can make one up. And in this case, we're going to use the letter B. Now, we've been told in this instance that mice who are black are dominant to white mouse for this colour trait. And so what we're going to do is, for capital B, we're going to represent this as the black coat versus our recessive allele, little b, will be a white coat. We've then been told the parent phenotypes. And in our parent phenotype here, we've been told that we have a white mouse crossed with a black mouse. Based on this information, we can now determine the parent genotypes. So a pure breeding white mouse is another way of saying a homozygous white mouse. And if we know that white is little b, we are going to write our homozygous white mouse as little b, or homozygous recessive. Our black mouse, on the other hand, we've been told is heterozygous, so they have our dominant allele and our recessive allele. They have a copy of both versions of this characteristic. When we now look at our Punnett square, so we start by drawing our square, we draw our checker inside, and we fill in each of these particular parents. So if we start with the white male mouse, we'll go little b, homozygous recessive, and then we move on to our black female mouse, and this is a heterozygote. Now that we've filled in our parents, we can go ahead and fill in these particular crosses into our Punnett square, and we end up with two particular genotypes. I'm going to write as a fraction here because, again, this is what the question has clearly asked us to do right at the start. So in this instance, 1 out of 2, or 1 of every 2 of our offspring are heterozygotes. 1 out of every 2 are homozygous recessive. And in our phenotypes, this means for us, again, that 1 out of 2 will be black and have a black coat, and 1 out of 2 will have a white coat. So this process here for writing out our Punnett squares is a really, really nice, easy way of making sure that you maximise your marks in an assessment.